Welcome guys. Now I will discuss about thyroid physiology, function and regulation. The main function of thyroid gland is to synthesize T4 and T3, and the only known physiologic role of iodine is in the synthesis of these hormones. The recommended daily allowance of iodine is around 120 microgram for infants, around 100 microgram for children, and 150 microgram for adolescents and adults. Whatever the chemical form ingested, iodine eventually reaches the thyroid gland as it is ionized form or iodide. Thyroid tissue has an avidity for iodide and it is able to trap with a gradient of around 100 to 1, transport and concentrate it in the follicular lumen for synthesis of thyroid hormone. To form thyroid hormone, trapped inorganic iodide must be organified onto tyrosine, residues of thyroglobulin in the follicular lumen, a reaction catalyzed by thyrotropin. Iodination of about 40 of this tyrosine forms monoiodotyrosine or diiodotyrosine. 2 molecules of diiodotyrosine then couples to form 1 molecule of T4. And 1 molecule of diiodotyrosine and monoiodotyrosine forms T3. Once formed, hormones remain stored as part of thyroglobulin in the colloid of the follicular lumen until ready to be secreted from the thyroid. In adults, the thyroid secretes approximately 85 microgram of T4 and 6 to 7 microgram of T3 daily. Only 20% of circulating T3 is secreted by thyroid, while the remainder, around 80%, is produced by deiodination of T4 in extrathyroidal tissue by iodotyrosinase deiodinase, type 1 and type 2. In the pituitary and the brain, approximately 80% of required T3 is produced locally from T4 by type 2 deiodinase. Although it is present in the blood, only about 1/5 the concentration of T4, T3 is physiologically active thyroid hormone because it binds the thyroid hormone receptor with 10 to 50-fold greater affinity than T4. When we see thyroid hormone function, entry of T4 and T3 into cells is facilitated by specific thyroid hormone transporters, and once inside the cell, T4 is converted to T3 by type 1 or 2 deiodinase, as I have said, and intracellular T3 then enters the nucleus and binds to thyroid hormone receptor. Binding of T3 to the thyroid hormone receptor causes recruitment of co-activator molecules, transcription of messenger RNA, and protein synthesis. So thyroid hormone functions in increasing oxygen consumption, stimulate protein synthesis, influence growth and differentiation. It also affects carbohydrate, lipid, and vitamin metabolism. It is critical for brain growth and development, and it potentiates the effects of catecholamines. Approximately 70% of circulating T4 is bound to thyroid binding globulin, and most of the remainder is bound to albumin and prealbumin, also called transthyretin. Only 0.03% of serum T4 is unbound and comprises free T4. Approximately 50% of circulating T3 is bound to TBG, and 50% is bound to albumin, and only 0.3% of T3 is unbound or free T3. When we see thyroid regulation, the thyroid is regulated by TSH, or thyroid stimulating hormone, a glycoprotein hormone secreted by the anterior pituitary. By binding to the thyroid stimulating hormone receptor on thyroid gland, it stimulates all steps of thyroid hormone biosynthesis, from uptake of iodine to release of thyroid hormone. TSH is a heterodimer composed of alpha and beta subunits. The alpha subunit is common to luteinizing hormone, follicular stimulating hormone, and the chorionic gonadotropin, while the specificity of each hormone is conferred by the unique beta subunit. TSH synthesis and release are stimulated by thyrotropin releasing hormone, a tripeptide that is synthesized in the hypothalamus and secreted into the pituitary. In states of decreased thyroid hormone, TSH and TRH are increased, while increased thyroid hormone inhibits TSH and TRH production. Although thyroid stimulating hormone levels can be measured in serum, circulating levels of thyrotropin releasing hormones are very low outside the neonatal period. Further control of the level of circulating thyroid hormones occur in the periphery. In many non-thyroidal illness, circulating T3 levels fall due to number 1, decreased extrathyroidal production of T3 by type 1 dehydrogenase, and the second is increased inactivation of T4 by type 3 dehydrogenase. These changes may be induced by factors such as fasting, chronic malnutrition, acute illness, and certain drugs. Although levels of T3 may be significantly decreased, levels of free T4 and TSH might remain normal. The decreased levels of T3 may be physiological adaptation in such situations, resulting in decreased rate of oxygen consumption, decreased rate of substrate use, and of other catabolic process. This is all about a summary of thyroid hormone physiology, function, and regulation. Thank you for watching.